Okay, well, hello from sunny Phoenix, Arizona. It's about 10:30 a.m. here. Um, I want to first thank Antonio Baines and the organizing committee for allowing me the opportunity to give this presentation. Um, it's a good thing I'm not traveling right now. Just about to run into a bunch of travel here in the next couple of weeks. So it's good to be able to do this from home or from my office here at TGen. A little background. So TGen is short for the Translational Genomics Research Institute. We'll pull the phone a little closer to make sure you can hear. Um, we were founded back in 2003, essentially a relatively, relatively large group of individuals who were at the National Human Genome Research Institute on the NIH campus in Bethesda, got together some ideas about forming a translational research program. And this was kind of before the word translational was even a buzzword, really trying to form a nexus between sort of what was happening in the field of genomics at the time and what was happening in the clinic at the time as we were starting to see the development of more and more targeted therapies, primarily in the area of cancer, but in other disease systems as well. Um, and so we all moved out here to Arizona, Phoenix, Arizona, about 2003 to set the institute up. We are currently in our 11th or 12th year um, of existence, and things are actually going pretty well for us. Challenges, just like everyone else has, but all in all, things are going incredibly well for us. These are very exciting times in the field of genomics medicine, and we're glad to be a part of it and kind of feel that like we are participating as thought leaders way out in the fringes of this burgeoning field. Um, so the title of my talk is um, bringing genomics to clinical cancer care, the promise and the challenges towards improvement in clinical management of patients with cancer. And I'm proud to be able to present this on behalf of all of my wonderful colleagues, both in the laboratory and in the clinic. Um, I guess before I get started, I just wanted to sort of walk you through a general outline. I'll probably start off with some slides that sort of lay the groundwork and framework for the rationale behind why we're doing what it is we're doing, why we think it's important, how we got here, um, and then move into some of the technical aspects of the methodologies and techniques that we're using and some of the background behind those, and then talk a little bit about moving those methods and techniques into a clinical setting, a clinical laboratory, CLIA labs, and the validation of these types of tests, and then walk you through some examples of how we've utilized this platform that we built out here to support our efforts in clinical genome sciences, again, primarily in the area of cancer. So hopefully by the end of the presentation, you'll have gotten a little bit of a background on the subject, some real life case studies and examples of how we're moving these things forward and utilizing them. And then I'll end with a few slides on, some of the things, additional considerations that need to be sort of kept in mind as we continue to move forward with these technologies in the clinic. Also, forgive me if I'm sneezing and snorting and coughing because I'm kind of in the at the kind of late mid stages of a pretty nasty upper respiratory tract thing that was part bacterial, part viral, and so I'm just trying to get over that. So pardon me if. Make sure if you can't hear me clearly, just flip me a note, and I'll try to get closer to the phone. So, this slide is pretty simple, but I think it's important in sort of recognizing the fact that cancer, at its most basic, in its most basic form, is a genetic disease. In many cases, there's some intrinsic or extrinsic factor that causes a molecular assault, usually genetically at the DNA level, which leads a cell from being normal and pushing it down the neoplastic pathway. Some of the intrinsic factors we can think about are inherited mutations and genetic predisposition. I'm sure there are probably some people in the audience who spend quite a bit of their time studying those types of subjects, whether it be genetic epidemiology or hereditary cancer research. And then just general aging. We know that there have been a number of studies in the past that show that there are just some general aspects of aging that might be related to the development of cancer, although many of those things are not necessarily all that clear. Extrinsic factors, of course, are many of the known carcinogens. We can think about the chemicals that are used in the production of cigarettes, UV irradiation exposure, and viruses like HPV and hepatitis that we know predispose to various forms of cancer. So the discovery of genes that are altered during the tumorigenesis pathway has led us to new and better understanding of the driving forces behind cancer and has provided new ideas for treating this disease. And some of the questions that are important, I think, from a health disparities perspective is, are some of these differences in population ancestry driving biology that impacts disease incidence and outcomes? And I think we're starting to see some interesting studies come out that are showing, I think, both sides of this from a health disparities aspect. My talk won't be focused on disparities, but I think it's important to bring that aspect up, that we're starting to see studies that are showing that biology may not necessarily be behind certain disparities. And I think one in particular would be multiple myeloma. I'm not going to talk about that, but we've done a lot of work in that disease space, where we show that, you know, historically African Americans have had higher incidence rates and higher mortality rates than whites. However, what we've done is we perform deep molecular profiling on multiple myeloma tumors from African American and European patients and show that there really aren't very many differences. And when we look at some of the indices, molecular indices that might define a high-risk patient or a patient who's likely to have a disease that will relapse and come back with a vengeance after therapy, we're seeing that those profiles are not enriched in African Americans or individuals of recent West African descent. And now that we're seeing better drugs being developed for this disease, like the IMiDs and the proteasome inhibitors, we're actually starting to see better outcomes in African Americans because we have better drugs. So from my standpoint, myeloma is a great example where the biological differences are not driving the mortality, have not driven the mortality differences. It was access to better drugs and better treatments for that disease and better distribution of those therapies and treatment options. I think on the flip side of that coin is a disease like pediatric ALL, acute lymphoblastic leukemia. Jun Yang at St. Jude performed a really beautiful study, was published in Nature Genetics, where he performed genome-wide association studies on a large series of children with ALL. And then he performed principal components analysis to essentially group individuals by their genetic ancestry. And what he was able to show was that children who had greater than 10% Native American ancestry tended to be diagnosed with a disease that was much more likely to relapse after treatment, much more likely. And the kids had much, much higher mortality rates. However, he was able to show that if you gave those kids a more intense dose of chemotherapy, which is called delayed intensification, that you could essentially remove the disparity. And so again, in that case, perhaps biology was playing a role in the outcomes. However, you can eliminate that disparity by giving the kids the appropriate treatment. So again, I think that we're learning more about biology and its role in cancer health disparities. And although my talk won't be focused on disparities, I thought it was important to sort of lay that groundwork and that framework as I move forward through my presentation. There are essentially two major classes of cancer genes that I'll be talking about today. There are some other classes like gatekeepers, but I'm going to focus on two classes. One are the oncogenes, and oncogenes are genes that, when they are activated abnormally or produced at levels higher than they should be in a particular cellular context, that activation of that gene will lead a cell down the neoplastic pathway. Some of the well-known oncogenes that some of you might have heard of are PI3 kinase, AKT, mTOR, KRAS, EGFR. Many of the receptor tyrosine kinases also fall into the class of oncogenes, and these genes become activated, and their activation can be associated with cellular transformation and the onset of tumorigenesis in cancer. The other, and one way to look at it, and these are sort of cartoons, is the oncogenes would be indicative of the accelerator in a car. So, of course, there are two ways to make a car go out of control. One is to stick the accelerator to the floor abnormally, and that's the oncogene. It's like the accelerator. The other is the tumor suppressor. The tumor suppressor is like the brakes. Tumor suppressors are genes that, when turned on, typically tell a cell to stop proliferating, stop growing, stop surviving. And if you lose that particular signal, the cell doesn't know when to stop growing or when to stop proliferating or when to stop surviving. And so these are the tumor suppressors. They're like the brakes. So again, you can cut the brake line as a second mechanism to make a car go out of control. Interestingly, many cancer genes will have both of these particular mechanisms occurring. So you will have the tumor suppressor that it's like cutting the brake line, and then you'll have the accelerator stuck to the floor where you have an activated oncogene, and those two signals can send the cell really fast down the neoplastic pathway. Now, or neoplastic highway. I think it's also important to note that in some instances, it may only take one event. I think cancer is a process. So there's normal, in many cases, there's sort of a next step of sort of pre-malignant lesion. And then that particular pre-malignant lesion then has a propensity to become malignant. And so many tumor types, we've seen the progression paths where you go from normal to pre-malignant to malignant. You can think about colon cancer, normal colon epithelium to adenomas, or polyps to adenomas to adenocarcinomas. Multiple myeloma, you have normal plasma cells, you have monoclonal gammopathy of undetermined significance, or MGUS, then you have smoldering myeloma, and then you have full-blown multiple myeloma. So many cancers, and then different events or genetic events occur along that pathway that make a cell jump from one particular step to the next. Also thought it was important to provide that context. We've learned a lot about the genes and the proteins that are involved in cancer, and through that knowledge have also gained considerable in-depth understanding of how these particular molecules play together within a cell. And as complicated as a slide like this may look, which is the KEGG cancer pathway, this is probably a super oversimplification of things. But it at least provides some standard understanding and framework of the events that can occur. We have the receptor tyrosine kinases. We have some of the important signaling components, such as the AKT pathway, PI3 kinase, AKT, mTOR pathway, or the RAS-RAF-MEK-ERK pathway. We also have cell cycle, apoptosis, Wnt signaling. There are a lot of these pathways and networks that, when they become perturbed, if one alteration occurs, it essentially sends a signaling path to tell that sort of abrogates the normal signaling, and that collection of events is what causes a normal cell to begin moving down the neoplastic pathway. So again, although we've learned a lot, we still have a long ways to go. There are a lot of holes and gaps in our understanding of how these different proteins interact with each other in a normal context or in an abnormal context, such as cancer. It's also important to know that we understand those genes, and we've also learned a lot about how those genes can become perturbed or altered in cancer. And it's important to note that different alterations can occur in these genes that can sort of provide a context for cancer, whether it be a gene like EGFR, HER2/neu, which is amplified and overexpressed in a particular subtype of breast cancer. The estrogen receptor can be overexpressed or mutated or be involved in oncogenic fusions. EGFR can be amplified, it can be mutated or overexpressed in certain forms of lung cancer. KRAS, we know that probably 99% of pancreatic cancers contain a mutation in the KRAS oncogene, which is its primary oncogenic signal. A lot of work has been done. Our group found oncogenic fusions in the FGFR2 gene in metastatic cholangiocarcinoma. And again, many of these are the oncogenes that are perturbed in cancer, just a subset. And then some of the tumor suppressors that are altered in cancer that can include P53, P10RB, CDKN2A, or P16. And these genes are typically deleted or mutated, or their promoter region is hypermethylated, which can also lead to inactivation or, again, cutting of the brake line. And then there are certain genes that are involved in fusions where chromosomes will break apart and fuse together abnormally. And then the fusion region between forms a chimeric protein or chimeric gene product and protein product. One of the more famous would be BCR-ABL, which is the result of the Philadelphia chromosome in leukemia. Tempus-ERG fusions that are very common and frequent in prostate cancer, and the EML4 ALK translocations and oncogenic fusions that occur in a subset of about 5% of patients with non-small cell lung cancer, which is also a known oncogenic event. Previously, it was important that we had to have multiple technologies in order to identify these events. For instance, to identify the HER2 amplifications in breast cancer, it's IHC, which is immunohistochemistry, which is performed in a pathology lab. Many times with EGFR, it's amplified. We can identify this at the DNA level using microarrays, overexpressed at the RNA level, again, using RNA microarrays, or mutated. And previously, we've used things like Sanger sequencing systems, capillary sequencing, pyrosequencing to identify these mutations, again, whether they're mutations in oncogenes or in tumor suppressors. But probably about 6 or 7 years ago, we saw the development and introduction of new next generation sequencing technologies that have really provided the opportunity for us to make all of these multiple measurements using a single system. And so they've sort of laid the groundwork for us to be able to perform very, very deep molecular profiling of tumors using a single experiment, so that we can make all of the appropriate measurements. And without going in too deep, one way to look at it is the genome consists of about 3 billion base pairs or letters. And just to put this in context, one sheet of paper, you can get about 5,000 letters on it, which equates to one ream being about 2.5 million letters. A box of paper is about 25 million letters, and you need about 120 boxes of paper to get to 3 billion letters, and I think that's about the height of the National Monument or something, I don't know. But the other important thing is that in order for us to deal with some of the error rates that can occur because of the chemistry and the mathematical modeling that takes place for genome sequencing, is that we have to shred it into small pieces, and then we have to oversample to overcome errors. So we don't want to just look at 3 million. We want to look at 3 billion. We want to look at 3 billion probably times 100. So just think about the amount of information that has to be generated using these particular technologies. But it's great because we have incredible computing now, much better storage, faster processing, that allows us to not only generate this particular information, but to analyze it effectively and efficiently as well. So we take each one of these fragments, we shred it into pieces, and then we find usually about 50 to 100 bases in length. And then we say, where does this 100 bases map? What's the highest probability in the genome for this 100 bases to map? And that's how we map these reads across the genome. Another way to look at it is it's taking one piece of DNA that's, let's say, about 400 or 500 nucleotides in length, and we only sequence the two ends of it. And so this sort of represents a sort of a pileup of what we call paired ends. So one read would be here, its pair would be here. Now we don't have the sequence in the middle, but the computational aspects allows us to know that this sequence pairs with this sequence, and they should be about 300 or 400 bases away from each other. And on average, that's what you would get. So each pair would map, and there would be the space in the middle. And then as you pile these up along the region, you can begin to sort of collect the information so that each base is represented multiple times by this data. And the other important thing is that if these two ends map to different chromosomes, it would also mean that somewhere in this region of the genome, there's a breakpoint that has occurred. And if you have multiple reads that give evidence for that breakpoint, then that would suggest that you have perhaps a translocation or some type of genetic or genomic alteration that's occurring between or within that particular region of the genome. And so not only can we get the information on bases, the base content of each read, we can also get information on structural content. Also, if we know how many times we sequenced or how much data we generated, we can also determine how much genetic material is present in that region of the genome. Cancers can amplify regions to make more of a gene product, or they can delete regions to cut out a tumor suppressor, to like cut the brake line again. And so using this one technology, we can get so many different pieces of information. Again, we can identify the mutations at the base level across the genome. We can deduce copy number changes, so amplifications and deletions across the genome. And we can also identify these rearrangements and translocations and inversions that can occur within a cancer genome. And importantly, if we collect RNA from the cells, we can also look at the gene expression profile, not only like microarrays, just looking at one probe, but we actually get the base information of the RNA as well. So we can get incredible in-depth, high-resolution genetic information using these technologies in a way that we were never able to do, let's say, 5 to 10 years ago. So these technologies provide a comprehensive framework for genomic interrogation, again, in a single experiment. So if I have normal DNA and tumor DNA from a patient, and I collect RNA from the tumor, in that one sequence run, I can sequence the entire genome, identify all the mutations, all the copy number changes, all the structural events, and perform gene expression at the base level, the exon level, the isoform level, and identify oncogenic fusions in a single experiment. So because of the power of these technologies, we're now able to ask a relatively pointed question. And as we continue to see, additional development in the pharmaceutical community as it relates to targeted therapies, where we have different tumor types and we have specific genetic alterations, and the development of drugs that target those alterations. Again, BRAF V600E mutation that occurs in melanoma is a biomarker for the drug vemurafenib. HER2/neu and the use of the drug trastuzumab or Herceptin in HER2/neu positive breast cancers, the use of the drug imatinib or Gleevec for the use of BCR-ABL fusion positive leukemias. TGen has played a role in some of these, the development of proteasome inhibitors with our partners at the Mayo Clinic in Scottsdale, and activation of the NF-kappa B pathway, as well as the development of vesmodegib, which is a hedgehog inhibitor for patients with metastatic basal cell carcinoma, particularly those that have patched mutations. So as we see, continued development of specific targeted therapeutics around very specific genomic alterations, we can begin to utilize these next generation sequencing technologies to ask the question, is molecular profiling actually a rational approach to increase the options available to oncologists for treating cancer patients? So instead of, once a cancer patient has failed standard of care, and the perhaps failed standard of care at relapse, the docs pretty much, or the oncologists, have a dilemma where they have to sort of think about what's the next option for that patient. In many cases, the standard of care is not related to a specific targeted therapy, although we're starting to see some of that beginning to increase over time with more and more of these treatments. I think the other part of that is we now have better technologies, more sensitive and specific technologies and biomarker development. But once a patient fails standard of care at diagnosis and standard of care at relapse, the docs then have the opportunity to utilize clinical trials. And, to be honest, a lot of times they aren't armed with enough information to determine what might be the best particular treatment option for that patient at that particular point in time. And so perhaps if we can provide them with very specific molecular information against a particular tumor, that can perhaps provide the doctor with key insights to determine what might be an appropriate therapy to use in that advanced setting. Now, I will preface these comments by saying there's still a lot of complications in our understanding. Maybe at the end of this, I'll get some questions about the word heterogeneity and clonality. It comes up time and time again. So you think about a patient with advanced cancer. In many cases, there's not just one tumor cell, clonal tumor cell, that's propagating throughout the patient. There are multiple tumor cells. In a lot of instances, there is probably a single primary clone that gave rise to the patient's full-blown cancer. But each one of those cancer cells is continuing to divide and propagate and collect new mutations, right? And so when you think about that, we need to try our best to address the entire complement of the cancer in the patient. Now, we've seen some interesting studies come out over the last year or two where individuals have taken a tumor, cut it into different pieces, sequenced each of the pieces, and shown that there are different genomic events in each of those pieces of tumor. We've also seen individuals take different metastatic lesions from the same patient and see the same thing. However, one of the things that we keep seeing, though, is that in many cases, the key driving events are there in all of the particular tumors within a patient or within a given lesion. For instance, there's a P10 homozygous deletion, and you see it everywhere. Now, you might see specific mutations in each one of those lesions, but all of them have the same P10 homozygous deletion, or they all have a P16 deletion, or they all have a KRAS mutation. So what we want to do is to be able to see if we can target those driving events, those quote-unquote driver mutations that are probably the mutations that have given rise to the primary cancer and are the primary culprit for tumor genesis. Now, there's another class of mutations that might be associated with drug resistance. So there are certain mutations that can occur that actually, if we have knowledge that mutation is there, it means that specific drugs will not be effective against that particular tumor. So we have to keep that context in mind as well. So it's a very sort of complex picture, but amazingly, we're starting to get a better view of it. I won't say that we have a particular high resolution view of it, but boy, we're starting to see it more and more clearly day after day. We're learning more, and this is an iterative process and an iterative system. We learn every time we do a patient, and we can apply that new knowledge to the next patient. So with that in mind, I'd like to provide you with sort of the framework that we've built out for our genomics-enabled medicine platform. And this is sort of an algorithm that just sort of has some pictures and a more general diagram of our process. There have been a number of papers that have been published that sort of demonstrate a similar process. There are a number of groups around the country and around the world that have developed similar platforms, whether it's the group at the Broad Institute, WashU, Baylor, just to name a few, and a number of companies that have gotten off the ground that are using these types of technology to distribute this more broadly across the oncology community. In many cases, they'll draw it as more of a linear process where you go from patient to, sequencing to a report. But I tend to draw it more as a cyclical process. And I used the word iterative a minute ago, meaning that we start with patient and consent and physician, and here's the green light go through biopsy, shipping and collection and shipping of the specimen, quality assessment of the specimen and the analytes that are derived of DNA and RNA or proteins, through sequencing, data processing and analysis. And then we've developed a clinical genomics engine that will take the genomic information from the tumor, match it to the targeted drug space, and then provide a report that's discussed at a molecular tumor board, which then provides information or recommendations to the treating oncologist based on the molecular information. And again, as I mentioned earlier, it's a cycle. It's an iterative process, meaning that when we finish with patient 1, we learn, and we can apply that knowledge to patient 2, and then to patient 3 and 4. So although this can sometimes be framed out as an N of 1 study, it actually, over time, you begin to collect cohorts, and we learn from the outcomes. So we want to be able to follow after treatment. How did the patient do? Did the patient respond? If so, for how long? If the patient relapses and a new tumor arises, can we collect or get a biopsy from the new tumor and sequence it so we can ask the question, did new mutations arise? Did the tumor evolve? Did a particular subclone now gain a new growth advantage to become the dominant clone that's resistant to the current therapeutic regimen? And so we can continue to learn from these studies so that over time, this knowledge engine continues to grow, by which the more and more genomic data we collect, we can match it to the improved and increased knowledge to make these reports as accurate as possible over time. And that's our primary goal, is to provide the most accurate information and most clinically relevant information to the oncologist, so that they can make what we feel are more informed decisions as they begin to treat these patients with these advanced cancers. So given that we've built this pipeline out, one of the important aspects is that if it's going to be used in clinical management, then it has to fall into the auspices of a clinical laboratory and clinical processes. I'm going to take a brief time out for a little bit of, just give me a second. I'm struggling here. Sorry, maybe I should have muted. Okay, so what we're dealing with here are what we would consider laboratory developed tests, or LDTs, and I bolded that for a reason. There was this sort of, I know these are CMEs, and there are some questions that are going to be asked, and I bolded a few things, so some of you might want to keep that in mind. So the laboratory developed test is a test that's performed in a clinical laboratory. In many cases, it's what we call home brew, which means that you've developed a very specific test to make a very specific measurement, which means you have to meet a certain series of validation metrics. It's also important to note that we all hear this word biomarker quite a bit, and what we're dealing with here, there are two different classes of biomarkers. If you talk to anyone involved in the clinical laboratory space, there's the integral biomarker and the integrated biomarker. In the case of an integral, this is where the data is actually going to be used for clinical management. So in a clinical trial, you screen tumors in patients for a particular mutation, and if they have the mutation, the patients get treated because of that mutation. That is an integral biomarker. Whereas by which an integrated biomarker is where you're doing more discovery, where you're treating patients, all of the patients with a drug, you're collecting tumors, you profile, and you're looking for differences in response. So the patients that respond versus the ones that don't respond, is there a molecular signature of response? That's more of an integrated biomarker where that information is not necessarily going to be used to determine how to treat the patient, but you still want to collect information for the development of a particular biomarker downstream. So that's the integrated. So what we're dealing with here are the integral biomarkers, where we're using molecular information that will actually be integral to the clinical decisions that are made by the doc. So, there's, two types of validation. I also think that's important. Specifically, there's technical / analytical, and I'm going to put a slash here instead of and. Technical / analytical validation, and this is actually required and regulated by CMS or the Centers for Medicare and Medicaid Services, and CLIA, which is the Clinical Laboratory Improvement Amendments of 1988. And so they actually regulate technical or analytical validation of a particular laboratory developed test. However, clinical validation or the clinical validity of a test is actually regulated by the Food and Drug Administration or the FDA. Now, more recently, the FDA have gotten more involved, and they actually want to be involved in the regulation of some of the technical and analytical validation as well. And if you want to develop a molecular device or have a real FDA-validated biomarker, you actually have to provide information on your technical and analytical validation, and it has to be done by their particular requirements and regulations, and that is the FDA. But in general, laboratory-developed tests are regulated by CMS and CLIA. And for these technical / analytical validations, you have to develop a test and validate it, and that validation process must provide information on the reproducibility, repeatability, sensitivity, specificity, precision, and accuracy for detecting or making a particular measurement. And in our case, it's identifying somatic events in a tumor. Somatic meaning tumor-specific alterations. So the alterations that have arisen in the tumor. We're trying to make those measurements, identify the amplifications, deletions, mutations, structural rearrangements, or gene expression differences that have occurred in the tumor. And we have had to develop a validation plan and execute that plan in order to show reproducibility, repeatability, sensitivity, specificity, precision, and accuracy for that particular test. And we've had to use a series of cell lines and normal tumor pairs of the type that for the test that we're making, and I'll talk a little bit about that later. And for both DNA and RNA, and on the DNA level, it's mutations, copy number changes, structural events, and to determine, for instance, sensitivity, we did dilution studies where we took tumor and normal DNA and we diluted them at different titrations to see how far can we go down in tumor content, genome content, to identify a particular genomic event like a deletion or a mutation. At the RNA level, we compared the RNA sequencing data from validation samples against Affymetrix gene expression data that have been generated in another CLIA lab to look at the correlation coefficients and how well do we match up with that Affy data, and use that as information for accuracy of our RNA laboratory and our pipeline. Interestingly, we also, one of the common events that occur in cancer are oncogenic fusions at the RNA level. I mentioned those, BCR-ABL, Tempus-ERG, EML4 ALK. And so in order to validate that, we actually just published a paper recently where we developed a series of synthetic oligospike-ins that recapitulate known oncogenic fusions like EWS, ATF1 that occur in Ewing sarcoma, EWS FLE that also occurs in Ewing's, the ML4-ALK, BSR-ABL, a bunch of known oncogenes. We created long oligo synthetics, cloned them into an in vitro transcription vector, made RNA, and then spiked it into cell line DNA so we could really study how well we can identify oncogenic fusions as our validation. We determined precision against a series of different analysis pipelines using different fusion detection algorithms, and generated a series of graphs that look at different fusions, and using different dilutions of the fusion in the cell line RNA. How well do we identify those fusions? And interestingly, we use 3 different fusion detection algorithms. And interestingly, if you look here at Tempus ETV1, you can see that only one of the 3 fusion detection algorithms picked that fusion up. And then here, if you look at EML4-ALK, one of them missed it completely, regardless of dilution level. So we were able to test different algorithms, different dilutions, to really validate our ability to identify oncogenic fusions as part of our validation. So in the development of this clinical laboratory, we also had to develop a series of standard operating procedures, or SOPs, such as laboratory quality assurance, specimen accessioning, so receiving samples, putting them into a laboratory info management system that's that's running under the auspices of HIPAA to ensure privacy, histopathological tissue qualification of the specimens, analyte extraction and qualification, analytes being DNA and RNA from whole blood, from frozen tissue, from paraffin-embedded archival tissues. We have several tests that are based on quantitative PCR and on next-generation sequencing, primarily using these platforms. We also had to validate our bioinformatics analysis pipeline and our reporting. So the report that we generate, either a technical report that gives the technical qualifications and metrics of a particular sequencing run and analysis, or an interpretive report that actually will match up a series of mutations with a series of known therapeutics that are related to that particular genomic event. And this is just a copy of our CMS license for the clinical laboratory. And for the FDA, this was a document that had to be submitted to the FDA for our validation for one of our clinical trials, which is a phase 2 study for metastatic melanoma patients that have BRAF V600E negative tumors, and we're sequencing the tumors to identify therapeutically actionable events and treating based on the molecular profile of these tumors. And this was the IND and the IDE document that had to be submitted to the FDA for approval to start that study. So it's a lot of work. We, at the DNA level, for a lot of our studies, we're taking a very comprehensive approach where we're taking more of a genome-wide view of the genome to capture all of the structural events and to deeply assess the coding regions around the exons to identify mutations with high levels of sensitivity and specificity, and also mRNA sequencing. So we're really focusing on the messenger RNAs because currently, right now, most of the therapeutically relevant information is coming from the messenger RNAs, although we know that there's a lot of work ongoing with microRNAs and long non-coding RNAs. But currently, those are not as clinically relevant, but I think over the next 5 to 10 years, they might be. So right now, we're just focusing on the mRNA. Again, using really high resolution, fast sequencing, this particular system will generate a very comprehensive genome and whole transcriptome quality data in about 30 hours. We have a really amazing analysis pipeline that was developed by Dr. David Craig, and then we have an amazing computational environment that's been developed in collaboration with Dell Computing to process data. Again, we have our somatic compendium report that will have the mutations and alterations that are present in a particular tumor. And we have a drug-gene relationship database that's been built by Jeff Kiefer and Sarah Byron here at TGen that matches up all the known pharmaceutical space and targeted therapies with their known biomarker information, either general rules or very specific rules. And then a series of heuristic algorithms have been put together computationally to match these lists together to generate a report, a clinical report that can be provided to an oncologist that will have the drug, the genomic information that's relevant in that particular tumor, the indication rather to give the drug or not given that genomic context, and a bunch of ancillary information that we can provide, and we can customize these reports such that some docs may want to know whether or not there are clinical trials available for that particular drug that their patient can go on. So I want to walk through a series now of, and I only have about 15 minutes left, so I'm going to walk through these pretty quickly, just some sort of case studies and anecdotal evidence of the process and how we've used it and situations where we've seen some really excellent outcomes in patients, and I guess these could be called exceptional responders. And we did a study in triple-negative breast cancer that was published about a year ago. So an aggressive type of breast cancer that lacks ER, PR, HER2, tends to occur more frequently in young African American women. And although 70% cure, 30% are chemo-resistant and very, very difficult to treat, we know that patients that have BRCA2 germline variants tend to be diagnosed with these basal-like triple-negative tumors. And there's some information suggesting that PARP inhibitors are effective in that setting. However, there are really no effective treatments for women with chemo-resistant triple-negative disease. This study was done as a partnership with Life Technologies and US Oncology, where we performed whole genome, whole transcriptome sequencing on about 14 patients with metastatic triple-negative disease to work through this process and identify therapeutically relevant information that can be provided to the treating oncologist. Again, this was published in Molecular Cancer Therapeutics. One patient in particular was a patient with metastatic triple-negative. We identified a BRAF amplification at the copy number level, and further deduction of this particular information showed that it was part of what we call a double minute, which is extracellular a circle that is made up of different bits and pieces of DNA from different chromosomes that replicates on its own. And using FISH, we were able to show that these were in fact double minutes. And so they're just pumping out tons of BRAF. So our report indicated BRAF, HRAS, and IMPP4B, which is a tumor suppressor involved in AKT signaling. We ended up suggesting or recommending a combination MEK-AKT, and you can see this is tumor before and after only 2 cycles on this combination. There was essentially what Joyce O'Shaughnessy called an otherworldly response in this particular tumor. Again, this is one of those settings where the patient would never have gotten this combination of therapy had we not provided that molecular context. We find this context in quite a few women with breast cancer, metastatic, triple-negative, as well as many of the metastatic ER positives. And we found lots of alterations in these 2 pathways, the RAS-RAF-MEK-ERK pathway and the PI3 kinase AKT and TOR pathway. Another interesting series of observations occurred in metastatic cholangiocarcinoma. The study was published earlier this year, where we identified several therapeutic contexts genomic contexts that led to relatively unique therapeutic contexts that led to interesting outcomes in these patients. Very difficult to treat tumor type. One patient had an inactivating mutation in a gene called ERRFI1. Importantly, ERRFI1 is a sort of endogenous inhibitor of EGFR signaling. So it sort of binds to the heterodimerization pocket of HER receptors, such as EGFR and HER2, and prevents them from dimerizing. And it's their dimerization that leads to activation. So if you lose ERRFI1, you get hyperdimerization and hyperactivation of EGFR. And in this study that was published by a group back in 2007 in Nature, they were actually able to show that if you knock out ERRFI1 or MYC6, you can abrogate EGFR phosphorylation and activity, and a series of mutants you can reverse that. So this is a very, very important tumor suppressor that, if lost, leads to the activation of EGFR. So the tumor board discussion was to use an EGFR inhibitor, and lo and behold, this is tumor before, tumor after, 60% reduction in tumor after only 2 or 3 cycles on an EGFR inhibitor. Again, this patient would never have gotten this drug had the doc not had that genomic information. This is sort of the metabolic activity looking at that tumor. This is before, and this is after, so they're shutting down the tumor activity almost completely. We also found novel oncogenic fusions and translocations that include the FGFR2 oncogene in 3 different metastatic cholangiocarcinoma patients. At the time, there were a series of FGFR inhibitors being moving through therapeutic development. One that was pretty close to FDA approval was pazopanib. In these patients, we went for FGFR inhibitors. And sorry, this should be corrected. But anyway, these patients went on FGFR inhibitors. This is tumor before. You can see the tumor is pretty much gone after 4 months. This is looking at densitometric measurements of the tumor before. This is after 6 weeks on ponatinib. You can see that the tumor is pretty much gone away. And here's tumor, and the tumor has gone away again after only 6 weeks. Patient would never have gotten an FGFR inhibitor had the docs not had that therapeutic contact. And so we're seeing a bunch of trials starting to open up, and we're hoping to work on one ourselves and have some proposals in the works to look at this disease. And I think it's important because we know that metastatic cholangiocarcinoma is a health disparity. It's much more common in African Americans. And so I would love to interrogate a series of tumors from African American patients to see if these genomic contexts are there, and if so, perhaps we can find a way to treat these pretty nasty tumors. Lastly, I want to talk about a young lady who happened to be diagnosed with neuroblastoma, really young girl. This is a pediatric setting. She happened to be African American. We sequenced her tumor, and these tumors typically commonly have, this is a neuroblastoma, they typically have NMYC amplification or mutations in ALK. This patient did not have NMYC amplification and did not have an ALK mutation. But we did find a rearrangement in a gene called PTPAD, which is a tumor suppressor. And here are some of the discordant read pairs that are defining this weird structural event occurring in PTPAD. Importantly, PTPAD inactivates the aurora kinase oncogene. And aurora kinase activation is very important for NMYC stabilization. So this was an interesting context in that this possibly was leading to NMYC stabilization without NMYC amplification. And so one way to look at this is phosphorylated aurora kinase leads to stabilized NMYC, which leads to tumorigenesis. And there is a known aurora kinase inhibitor that was in development by the pharmaceutical company Millennium. And so one would think if you could abrogate phosphorylation using this particular inhibitor, it would lead to a reduction in tumor and possibly a response in this patient. The patient did go on this drug. We were able to get the drug through compassionate use. And some animal models had shown that if you use this drug in certain pediatric tumor mouse models, that you could get very significant tumor reduction and increase in survival in these animals. And so the tumor board suggested the use of this aurora kinase inhibitor. She did get the drug, and she is having a great response. I was trying to get some images from my colleague at Children's Hospital Orange County, but couldn't. But it's just an amazing story where this young lady had a really nasty tumor, and she's on a drug that she would never have gotten, and she's doing well. So we're really excited about that one. Sort of just wrapping up, I think it's important. Again, CMS, CLIA, FDA requirements and guiding principles are driving the development of these LDTs, and we have to keep in mind the difference between the technical versus clinical validations. Also, clinical trials using FDA-approved and/or investigational agents. One of the important aspects for our gene-drug relationship database is the actual pharmacopoeia that's we're using. Some docs can only use FDA-approved drugs, so we have to use a database where our pharmacopoeia is selected only around FDA-approved drugs. Well, whereas other trials like our Stand Up to Cancer melanoma study, we're using investigational agents as well as part of our pharmacopoeia. So that all has to be taken into context. The other important aspect is access to these pharmaceutical agents, and we're seeing a lot more sort of compassionate use where we have a great context. There's no clinical trial, but we believe that a particular company has a drug that would work great in that context. The docs can write the company and ask for access. And it just really depends on where the drug is in the pipeline, where the drug is in its FDA approval process. And we're seeing more and more we're getting access. One of the big issues is combinations, right? Many of these tumors have more than one mutation. And we need to hit multiple pathways, RAS, RAF, MEK, or PI3 kinase AKT. These combinations are tough sometimes because of toxicities. And a lot of times there has to be at least a phase 1 dosing study that's been completed to know what the minimum tolerated dose for these combinations is. And so we have to, we're working through these combinations. One way to help us is the use of patient-derived xenograft models where we take a biopsy from the patient, implant it into an animal, and now we have the tumor growing in an animal. Then we can test these combinations on the tumor in the animal model and ask whether or not that combination is very effective against that tumor. And then perhaps we can work towards moving into a clinical trial for that combination. And lastly, and I won't get too deep into this, but this is a biggie, which is reimbursement. How do we get this paid for? Are the companies going to provide some of these drugs compassionate use without cost? If it can be paid for, will the insurance pay for it? What are the mechanisms by which we can get the reimbursement? What evidence is required by the insurance company in order to get access to reimbursement? So that's a big issue that we're trying to work towards as it relates to continually pushing these things towards more broad and widespread distribution. And so finally, another big question is, are we going to be able to sort of retrofit this type of high complexity testing into the current clinical paradigm, or are we going to have to rethink the whole thing, including how we do the testing, how we do drug development, how we design clinical trials, how we educate the greater medical professional community, how we measure outcomes and standardize the nomenclature and the clinical lexicon, the whole aspects of reimbursement, and then education for patient and community outreach. So in closing, the promise of these technologies is to deliver the most appropriate and accurate drug information based on tumor profiles to hopefully get to the most effective outcomes and best outcomes. But there are a lot of challenges that remain, and so one of which is, will this approach lead to improved outcomes and effectiveness without sacrificing efficiencies to the current healthcare system? And making sure these technologies, if successful, are affordable, broadly distributed, and available to all patients. So with that, I'll close and put up an acknowledgement slide. Key acknowledgement is Dr. David Craig, who's Chief Bioinformatician here and Deputy Director of Information Sciences, who's just been incredible, my sort of partner in crime in developing this platform. Our CLIA laboratory group, some of our collaborators in industry, and our clinical collaborators at Mayo, Scottsdale Healthcare, Carmanos, and I put in the pediatric case at the last minute, and I'd also like to acknowledge Lenny Sender at Children's Hospital Orange County. So with that, I'll stop, and I think there was a minute or two to answer a few questions. I'll hang around. I do have to run off on a phone call in a minute, but I can hang around and answer a few questions, so thanks. Hello?